Emília Cunha from IPMA will present IMTA's environmental contribution now, and she has a pre-recorded presentation which I will now launch.
Work Package 5 dealt with IMTA environmental contribution, and the objective was to demonstrate the environmental advantage of IMTA. Since there were several pilot actions being tested in Integrate, we started by developing a conceptual model that would incorporate those pilot actions. The conceptual compartment model ended to be basically a model of saltwater pond IMTA, and the environmental performance of the system being carried out was evaluated through a life cycle assessment. Let's start with development of the conceptual model. 5 pilot actions were considered in the modeling. 2 were carried out in laboratory conditions, such as the pilot action of AgroCampus and the pilot action of NWIG. AgroCampus had a pilot action with 4 compartments. Primary producers, in this case phytoplankton, that were used to feed filter feeders, oysters, whose pseudophysis fed the composer, the tetrivores, as sea cucumber. The nutrient-rich water from those compartments was used to grow other sort of primary producers, macroalgae. The water was finally released into the environment. NWIG has a pilot action with 3 compartments. It was a RAS system with fed carnivores, lumpsucker, primary producers, macroalgae, and decomposers in a bacterial biofilter. The 3 next pilot actions were developed in the semi-industrial to the industrial facilities located in the coastal zone on tidal onshore areas. Alga Plus is a small and medium enterprise with a pilot action based on the company production system that is composed basically by 2 compartments. Fed carnivores, sea bass and sea bream, and primary producers, in this case several species of macroalgae. The nutrient-enriched water from the carnivores is used to grow the primary producers, the macroalgae. The water was finally released into the environment. Setaqua pilot action took place in a large facility of a decommissioned salt industry company in the old Esteros. The pilot action had 3 compartments: fed carnivores, sea bass and sea bream, primary producers, phytoplankton, and filter feeders, oysters. In the same pond, the nutrient-enriched water from this pond was used to grow primary producers, macroalgae, ulva and porphyra. The water was finally released into the environment. IPMA's pilot action was the other pilot action. It took place in an aquaculture research station. The system was composed by 5 components: a polyculture of fed carnivores and detritivores, filter feeders, and natural producers as phytoplankton. The primary producers, macroalgae, mainly ulva S species, were produced in a separate pond with the water pumped from the fish and oyster ponds. The result of conceptual compartment model developed for land-based IMTA in ponds accommodates 5 trophic compartments: carnivores, detritivores, filter feeders, phytoplankton and macroalgae, and displays their structural organization and function. The forcing function is pond management. A life cycle assessment of pond IMTA was carried out to identify the environmental impacts of the semi-intensive system developed by IPMA and the semi-extensive by Setaqua. The objectives were to determine the environmental advantages of these systems and recommend actions to minimize impacts. The assessed system was the farm itself. The inputs in the production, fry, feeds, energy, equipment, and the rubbish associated. In these boundaries, it is included fish fingerlings and oyster spat bought in hatcheries and transported to the site, the posterior farm operation with seeding of the ponds, diverting the water to the macroalgae production ponds, fish feeding, aeration, macroalgae and fish harvesting, and then finally fish slaughtering. Packaging and transport to the retailer was not included. The functional unit to which all inputs and outputs of the analysis were related was defined as 1 ton of live oysters and fish produced. General data concerning the study system are represented in this table. The main differences in the inputs were the size of the farm, the fish biomass that was much higher at IPMA's experiment in comparison to Setaqua, 50 to 25 times higher in the high density and low density SME intensive system in comparison to the SME extensive in the case of fish. The amount of water and the amount of feeds. As for the outputs, the fish and oyster biomass gain was higher in the SME intensive systems, but the FCR was very poor. Macroalgae biomass gain was higher at Setaqua. The impact categories used in the assessment were global warming, that assesses the impacts of gaseous emissions, carbon dioxide, methane, and nitrogen dioxide basically. Net primary production use, that refers to the use of net primary production as a biotic resource. Total cumulative energy demand, representing the use of fossil and nuclear energy sources in the system, from the input factors to the output. Acidification, referring to the negative effects on soils, ground and surface water, and ecosystem of acidifying pollutants. Eutrophication, it refers to the potential impacts of the high levels of nutrients in the environment, mainly nitrogen and phosphorus. Land competition, corresponding to the ground surface used to the production system. Water dependence, corresponding to the quantity of water flowing into the production system. On the study, we considered the following 11 processes and the relative contribution of the process to the impact categories show that the most demanding process were feeds. A process that contributes to most impacts being mostly important in net primary production use and global warming for the three systems. Direct farm activity was important in land competition and water dependence and driven by each of the three production systems also. Fish rejects contribute to eutrophication in the three systems, but while the effect was positive in the case of the semi-intensive systems, it was negative on the semi-extensive systems, implying that this was last system extracting nutrients from the environment. Water concept affect only water dependence, while energy consumption was important in the semi-intensive system, mainly the high density. Comparing the environment impacts of the IMET production systems, we can observe that global warming is the emission of the greenhouse gas to produce 1 ton of fish were relatively more important in the high density system than in the low density. Setaqua system contribution was the lowest. Feeds were the main responsible for those values. The total cumulative energy demand and global warming were influenced by similar processes, although energy consumption was more important in total cumulative energy demand in comparison to feeds in global warming. Energy consumption was the highest responsible for total cumulative energy demand in the semi-intensive system. Net primary production use showed that the production of 1 ton of fish and oysters was costly in Setaqua semi-intensive production system. Feed and fry were the only process that contribute to net primary production use, with feeds being responsible for most of the carbon produced. The highest acidification potential was observed in the high-density semi-intensive, but it was not far from the low-density semi-intensive system. Both were much higher than Setaqua semi-extensive system. Rejects fish were the main drivers of the high acidification potential of those two semi-intensive systems that is related to the higher FCR of those systems. Feeds was the second process responsible for the acidification potential and the main contributor from Setaqua semi-extensive system. In relation to eutrophication potential, the high density was the system that most contribute to eutrophication, followed by low density and Setaqua. Rejects fish and feeds dominate the direct emissions of nitrogen and phosphorus to the water. This was mostly noticeable in the semi-intensive system due to the highest FCR. Although there was a nutrient uptake from the environment by the fish, oysters, and the algae biomass produced, the amount of nitrogen and phosphorus rejection were still positive in the case of the semi-intensive system due to the amount of feed that was given to the fish. In the case of Setaqua semi-extensive system, the amount of biomass produced by the system and the low amount of feed delivered to the fish resulted in a negative total rejects fish. The land competition is the ground surface used to produce 1 ton of fish. This was more important in the high density system, although not much higher than the low density, while Setaqua used half of the land. Although the farm of the semi-extensive system occupied a much larger area than the semi-intensive, the fish were reared in a confined smaller area. Direct farm activity was similar in low density and Setaqua system. The amount of feed given to the high density semi-intensive system was almost double in the low density system, and that was reflected in the large feed's contribution to the impacts. The water dependence to produce 1 ton of fish and oyster, the high density intensive system required more water than the low density. Setaqua semi-extensive system required much less. For every system, water dependence was mostly associated with the rearing of the aquatic products, and therefore water consumption and direct farm activity were the most important process, with almost similar proportions in the case of the semi-intensive systems. Contribution of each rearing system to the impacts considered in this analysis for producing 1 ton of fish and oyster show that eutrophication, acidification potential, and total cumulative energy demand showed the largest disparity among the semi-intensive and the semi-extensive production system. With exception for net primary production use, Setaqua semi-extensive system had lower contribution to the impacts. IPMA's high-density semi-intensive system, in this case HD system, represents the highest impacts. To conclude, the present assessment is an estimate representing the range of potential impacts of semi-extensive and SME-intensive pond IMTA. Feeds is the largest single contributor to all environment impacts associated with pond IMTA production. The metrics feed gain, reads FCR, and water renovation energy consumption explained most variation regarding the environmental impacts of pond IMTA. Downstream macroalgae production decreased eutrophication impact. Improvements in environmental impacts could be brought about by on-farm improvements in feed gain, lower FCR, and that's for all impacts, water renovation, energy use, global warming, and acidification, that's the base impacts, improving feed composition and management, reducing energy consumption under current FCRs, and increasing downstream production of macroalgae. And thank you.